thanks everyone for coming here to listen to me speak about this kind of an unusual subject. Unusual in the sense that marine scientists don't traditionally think about translational sciences. And so what I'm going to try to talk to you about today is just get your feet a little wet. You're going to learn a little bit about what the translational paradigm is. You're going to learn how basic science translates to applied science through applications and development, and finally into what's called a public-private partnership, where you end up working with businesses, industries to create products, processes, or new idea data sets to help with the general human condition in the United States. Now, why is this important? Well, one extremely important reason is that Congress is talking about what is our return on investment in any federal grant program? What do we get for the work that I do on red tide killing little fishies? What benefit is there to looking at damselfish tumors? What does that mean, you know? And so as we talk about this, I just want you to kind of let your mind roll, because of, and avoid the person behind the green shirt over here.
I'm sorry, I'm trying to find the lights for you, which...
What are you looking for, the lights?
Here we go. There we go.
Okay.
Keep going. My apologies.
Okay, it's all right.
It'll look better.
So we're going to talk about translational science. Here's where I come from. University of North Carolina, Wilmington. This is the Center for Marine Science campus. It's actually the Crest Research Park. It's the campus for research, entrepreneurship, science, and technology. This particular campus, all 200,000 square feet of it, is new since 2000. We've been building a building of about 70,000 square feet every 4 years. The three buildings, the buildings I want to talk about tonight, first building is this one here, this large C-shaped building. This is a building, the first building that went up. This is the original Center for Marine Science. It's also the academic research and development space. These are the basic scientists in marine science, the people that look at sponge filtration, the people that look at oyster hatcheries, the people that look at how do fish grow, folks that are looking at climate change, people that are looking at water chemistry. The second building, this building was built, completed in 2000. The second building, here's an operations wing. This is for all of our ocean-going gear in our ships. It's where all of our physical oceanographers work. It's where we fix gear. It's where all of our ocean-going instrumentation is maintained. The third building, which is just outside, actually it's underneath the U here of UNCW, is a 16,000 square foot experimental shellfish research hatchery. State of North Carolina came to UNC Wilmington and said, you guys do most of the oyster and clam research in the state. Would you accept a building and develop best practices for reestablishment of the oyster industry in North Carolina? Well, I said, well, let me think for a minute, sure. That was in 2009. At the time that building was just being dedicated, this building here, the Mar Bionic building, Mar Bionic Marine Biotechnology in North Carolina, that building was just beginning. Like the behemoth building that you have outside, this was an ARRA construction project from the federal government funded by NIST. In fact, Mike Schmalley and I were talking, we had the same federal construction analyst who was at both of our campuses. He'd come to my campus and say, oh, you can't believe what's going on at Miami. He'd come here, Mike tells me, and say, oh, you can't believe what's going on at UNC Wilmington. Well, at any rate, this is where we do our work. We're 6 miles from the main campus of the university. Sometimes that's really good, sometimes that's really bad. Most it's really good. We are this close to the Atlantic Ocean. This is the Atlantic Ocean out here, Mason-Borough Island in between, the Intracoastal Waterway here. This dock is about 3 times the size now that it was when this picture was taken. I have a little over 100 faculty members, about 200 graduate students, 400 or so undergraduate students. And in this particular building that I'm going to focus on tonight, the Mar Bionic building, there are 22 investigators along with their staff, as well as industry. This is a building that was constructed by NIST as a public-private partnership building. So if we're carrying out all of our basic science in this part of the superstructure, it is going across this little bridge to this building where the translational science takes place. It takes place by first interacting with our applied and developmental scientists and moving it then ultimately into industry. We have 6 different industries in the building right now, ranging from skincare to pharmaceutical development to, of all things, wine. Now, you might ask why wine? The company's called Next Glass. Google it. They have an app, and their app, by taking a picture of any wine in any store, you can actually learn about that wine, and if they don't have the wine that your favorite wine is, they'll tell you another wine that's very similar to it, okay? They're one of North Carolina's 10 top companies this year, and we help them develop it's called LC coupled mass spec. It's a large instrument that costs a lot of money, and you shoot little amounts of material in it, and you get these great pictures. It allows you to compare all sorts of things. Okay, so that's really kind of the thing that we do. Tonight we're also going to talk a little bit about serendipity. Now, I have colleagues that say, no, it's not blind, dumb luck. And it's not blind, dumb luck, no. It's basically making discoveries that you weren't looking for. So as we go through this talk, just think about, oh, they hadn't planned that, and here's what they did with it. Because if you understand why things happen, you can always figure out a way to use them for some useful purpose. Now this, of course, serendipity comes from the ancient Ceylon book on the 3 princes of Serendip who were traveling around finding things they weren't looking for. It's quite an interesting story. If you haven't read it, don't try to read it in the Sanskrit. It's available in a number of different languages. The 2 things I plan to talk about in detail tonight are, first of all, the beach to bedside paradigm. This is the discovery of Brevenal, which is a cystic fibrosis drug that comes from Florida red tide. Okay? The second one I'm going to talk about is a molecule called Escortin. Escorts. These are molecules that actually carry drugs across membranes, and by modifying the escort just slightly, you can deliver the drugs to the point of action, to different parts of cells, to different types of cells. So we'll talk about those kinds of things. All of these happen just by not saying you had a failed experiment, but indeed saying, gee, there's something we can make of this. So the translational science, we're not going to talk about this, but if you Google translational science, you'll get the translation of words from one language to another. I guess there's a little bit of that, but that's not what we're going to talk about. We're going to talk about translational science, which is a cross-disciplinary scientific research paradigm. And it's motivated by the need for practical applications that help people. So when Mike talks about translational science, and he said the health aspects, that's kind of what we'll talk about tonight, but indeed, we are also looking for applications for other things we develop. Example I won't talk about in detail. We have developed an instrument that's diver held that you can carry underwater, put it on a coral, touch a coral, and learn something about the health of the coral without damaging the coral. So in the days of climate change, whether you believe it or not, temperature change in the oceans, ocean climate, ocean current changes, if those currents change, a lot of the ocean science community is interested in what's the water chemistry like, how are the currents changing, what is that doing to local climate. We're interested in what's happening to the living creatures that are in the ocean. And that actually was a reconstructed idea from a toxicology meeting I went to where there was a company talking about measuring cells and the pH and P and the oxygen consumption and the CO2 generation. And we said, look, if we turned that upside down and did this and this and this, we could create an instrument. That happened. That was kind of the disruptive technology portion of this. And then incrementally developed one that would go to 40 feet, to 80 feet. We're now at 200 feet. We're now redesigning to go from little mini computers to iPads and ultimately to iPhone. I'd like the iPhone 6 in there, I think, if we could do this. And the apps are very powerful, okay? So this is the kind of thing that we're actually working on. Now, how does that happen? It happens when groups of people interact together. It's an intercultural thing. So if you talk about a laboratory, and I don't know how many of you know what laboratories are made up of at different universities, but there's generally a principal investigator, the senior, the big guru, the one that has to deal with all of the other issues. There may be postdocs that are under that senior leader who are working with them. Under those postdocs, there may be graduate students, and there may be undergraduate students gaining work experience. So let's say that triangle exists in a laboratory and there's 12 people in the laboratory. Whose idea or whose opinion is the most important? Anybody? All of them. All of them. Undergraduates in our laboratories come most frequently with totally unbiased view of the world, other than, gee, we ought to make an app for that. I mean, this is what, you know. But the point is that the unbiased can give you a truly objective point of view on what it is you're trying to do. And so when we talk about the idea can come from anyone. There is this iterative discussion that goes on and on and on and on, and it should keep going on all the time. There's then experimentation which follows that iterative discussion that says, here's what we think we need to do. Can we experiment and prove or disprove our hypothesis? Generally, scientists are supposed to try to disprove their hypothesis, because if you can't disprove it, it must be true. There's no way to prove a hypothesis because you may have not done the right experiment. When you finish with experimentation, you move to product or process. So you think about, I don't know, a gorgonian that you're culturing in the laboratory. You find something that, gee, it makes your skin soft on your fingers when you touch it. So you look at that a little bit, you try to extract the material, you try to develop it. Once you've got the material, the pure chemical, you say, wow, there's the structure. Can I synthesize that? Can I get it from the natural source? What can I do with that? And then ultimately you might put it in an Estee Lauder anti-wrinkle cream, which is what actually happened somewhere. The important part is that this is a basic to applied to translational science paradigm. It's not always the same scientists that are the basic scientists, who is the applied scientist, who's the translational scientist. You don't have to be all three. I have basic scientists that tell me, I don't want to work with you on this stuff because I just want to do my basic science. And really what I ask them is, if you want to do your basic science, and I can give you a different revenue stream in addition to the grant revenue stream you have, by sharing your idea with an applied scientist to move toward a product, would you do that? Then most of the time it's, yeah, okay, but it's kind of dirty. I said, no, it's not dirty. This is the paradigm that's expected by the federal government of federal grant monies, giving something back to the American people as a return on investment for their investment in your research. So, after all that, now I'll talk to you about the subject of the talk tonight. So my talk tonight is to talk about two things. First is Brevenal, and this is a treatment of mucociliary diseases. These are pulmonary diseases. They're things like cystic fibrosis, COPD, black lung disease, asbestosis. You think if you can inhale it and get it stuck in your lungs, this is something that we're developing a therapeutic toward. We'll talk a little bit about its discovery. We'll talk about the therapeutic receptor site. They all have to have targets in order to be active. We'll talk about the target. We'll talk about how you deal with animals to see that, in fact, you're not going to ultimately have problems in humans when you administer the material. Worst thing in the world is to find a great drug and hurt somebody. First, do no harm, my physician friends. We'll talk about the preclinical tox and then moving toward discussions with the FDA about getting it into the clinic. We're very close to that right now with that drug. Second one is brevicin, and brevicin is also called the escortins, the things that carry things across membranes. We'll talk about what kinds of things we have to do. This is much earlier in the discussion and in the discovery phase right now. We'll talk about first putting fluorescent materials on our escorts and watching them go across membranes. We'll talk about measuring the uptake into cells, how you do that. Talk a little bit about generalizing the principle to more molecules. Talk about how you link them so that you can get them into a cell, cleave them, and have their activity on a cancer cell, for instance. And then talk about some future work. Reminding you, translational science is inherently interdisciplinary. If you don't have three different disciplines talking to one another, you haven't really got a translational science program. In fact, most of our work, when I was at Rosenstiel, we began this paradigm. I had a chemist, Mike was telling you, on 3 campuses, we brought people together. We had a grant funded to look at some of this early work with a chemist from the main campus, a pharmacologist from the medical school, and me, a biochemist. I like to call myself a marine scientist, but I've never taken a marine science course, so a biochemist. And you know, it's like those jokes, right? So the 3 of us sitting around in a circle on a Friday, we met every Friday afternoon. We'd sit in a triangle, can't sit in a circle with 3 people, sat in a triangle, And the postdocs and the graduate students were around the outside, and we would argue definitions. What does this mean to you? Oh, it means this. No, it means this to me. It means this to me. Coming to a common set of vocabulary so that we could discuss the research. Most of the postdocs and the students around the outside would listen and take notes and sit. And after about 3 months of that, we said, look, if you ever want to get down into the inner circle, you have to participate. So the next meeting, we're talking back and forth about definitions, and someone from the back, it was one of my postdocs, said, well, here's what I think that means. Come on down. Moved her right into the circle, okay? That's how it becomes important. You're creating a culture of interdisciplinarity. Is that a word? I don't know, a culture of that. And so the idea is that we do this all the time. We do this all the time in every other facet of life that you have. You have the family discussion around the dinner table. That's a discussion about translating what each of you know into what everybody else knows. Okay. The base material that I work with for all of the work that we're going to talk about tonight is this culture room. These are 10-liter bottles, 2.5-gallon bottles, stand about this tall, about this big around. There are 2,000 of them. Each of them contains 10 liters of culture media with Florida red tide growing in it. This is Florida red tide. It's a dinoflagellate, 2 flagella, swims through the water. It's photosynthetic. It is one of the most toxic single-celled organisms that's been described. It produces in upwards of 10% of its dry weight in brevitoxins, neurotoxins that cause lethality in the fish community and cause humans to bronchoconstrict and have pulmonary problems if the seashore, or if the waves break and they aerosolize the material. I have 3 technical staff that do nothing but maintain this culture room, purify material, and package it for sale to the retail community, the wholesale community, and for our certified standards for our work. We are moving from this type of culture very quickly now into 1,000-liter biofermenters that are all computer controlled that will generate the material for us. So this is the equivalent of about this shelf, this shelf, this shelf. 4 of these big shelves is one of these. We have one right now just to try it out. Arrived from British Columbia 3 days ago. We sat on a Skype interview with an iPad for 2 hours as we assembled this thing, 4 of us, and they're laughing on the other end of the line, you know. This particular type of an organization and type of a piece of equipment is going to be needed for clinical amounts of all of these drugs. So we have to produce clean material for the clinical community, and that's our job for the corporation who has licensed this material from the university. Just going to run through this really quickly. You start out with extraction with a solvent. We use chloroform usually, but we're moving to some other solvents now to get a crude extract that looks like this. It's kind of a brown, gooky material out of those greenish cultures. Dry that down, evaporate it under vacuum, and then start separating. You see this little green layer on top and the brown layer on the bottom? Green layer gets thrown away. Move to the next stage. Dry that down. Every step of the way, we're trying to get rid of things that are not what we want, only keeping the pieces that we want. By the time we get to a 4th stage of purification, you can see we are starting to resolve into multiple different colored materials, each of them composed of multiple numbers of chemicals that all have to be separated. Now this organism probably, I think the average they say that any living cell produces is about 60,000 different materials. My instruction to the group at one point was, let's just break it apart into all its component pieces and see what's active. Are you nuts? Well, anyway, we'll talk about that in a little while. I was a little nuts. And then a variety of different types of instrumentation. Now you can see, now look what's starting to happen. We're getting to purer compounds. You see some that are almost clear, some that are still brown, some that are still kind of cookie. Go through a couple other flashes, end up with this kind of a material. One more HPLC. This is what we get at the end. 3 different purified materials. I think you would agree that they're pretty white. They're pretty clean. They're all single materials. They are all of the brevitoxins, the things that cause Florida red tide poisoning, the Brevenal, the cystic fibrosis drug, and the Escortins, all in separate fractions. So from this single organism, by one purification scheme, we produce at least 15 different purified pharmacologic agents. 15 drugs, if you will, all that have different uses. Here's an idea of how we maintain inventories. So all of the different molecules that we either have natural from the culture or that we derivatize, in other words, make chemical changes to them and hold them, are labeled, categorized, and used by researchers around the world. We ship to Japan, to China, as well as many places in the United States, and then also for our own use. Our job as scientists funded by NIH, part of our job is to take those discoveries and get them out into the research community as quickly as we can. The reason for that? Somebody's going to make a discovery with that, and we want that to happen, again, for the return on investment to the NIH. Okay, so from beach to bedside, the discovery of Brevenal. Brevenal was a molecule. It's part of the Florida red tide. When Florida red tide comes onto beaches, this is what people look like. They cough and sneeze. Asthmatics are more sensitive. Elderly a little bit more sensitive. Certainly patients with any kind of a pulmonary disease should stay away from beaches when there is a red tide. Normally Florida's beaches are like this. Everybody's just excited to be there. This is a French woman. This is a local woman, and you know, French women come down here for the winter, no red tide. It's in the summer when we have our difficulties. Oops. Too quick. Brevitoxins do three things. They're neurotoxins. They cause neurologic poisoning in organisms that inhale them. They're particularly susceptible in lung. They cause changes in immunity, and they cause changes in cell metabolism. Those can all be exploited for drug use by just merely modulating the structure of the molecule to change the molecular, or to change the mechanism of action. Big slide, don't need to know all of it other than red tides occur here. They get blown ashore in wind. They come on the beaches. They can get blown 3 to 5 miles inshore and still cause bronchoconstriction. They will light or deposit on sliding glass doors, that white film, and if you rub that and then forget and rub your eye, you will develop a lacrimation syndrome from the toxins that are present. For the first 5 years that we had our large program project, and Mike talked about the centers, this is another project that was between us, I'll show you who all these people are, but a lot of different universities, all would converge on Florida when a red tide would occur in Sarasota. Boom, 15 people from around the country. We'd set up our laboratories in the closest hotel rooms on the beach because nobody else wanted to be there. You get them for 20 bucks a night. It was pretty good. And we would have Molten Marine Labs come and do all of the air sampling and basically break the whole thing down into its component parts. Here's the group. Group was composed, University of North Carolina, Wilmington, my laboratory led this for the NIH. We had components of the National Institutes of Health, the CDC, Florida Department of Health, all intimately involved working on the project with us. We had the Florida Department of Environmental Protection, who was very interested. Florida Marine Research Institute, who does all the red tide work on the west coast of Florida. Harbor Branch Oceanographic Institution for cruise data. Molten Marine Laboratories did all of the stuff that gets into the air. South Florida Poison Control Center was interested because they were doing reporting. University of Miami School of Medicine, and I leave Rosenstiel in that along. It was Laura Fleming moving back and forth from the two institutions. And then finally, closest colleague, Mount Sinai Hospital Research Institute, who actually decided that these were actually treatments for cystic fibrosis. And some pictures of all the people. I show this guy right here. This is Tom Mende. He was a professor in the Department of Biochemistry when I was a graduate student. He was my major professor. I started working with him in 1973. Worked with him from 1973 to 1977. When it came time to pick a research project, I said, I want to work on blue ring octopus venom. And he said, no, I think we're going to work on Florida red tide. I think we can have an impact there. Okay. He was right. Okay, here are the molecules. These are pulmonary toxicants. They cause severe bronchoconstriction. They reduce tracheal mucus clearance, so they basically make mucus thicker and doesn't expel as quickly. They reduce whole lung clearance, so that once toxins get in your lungs, they stay in your lungs longer. And these effects are in the picogram per cubic meter of air range. Now, how small is a picogram? Xanax takes 600 picograms per pill. This is 1 picogram per cubic meter of air. Very, very low concentrations. 2 different backbones. A long one, kind of like bananas. They're called ladder frame polyethers. I think you can see why they're called ladder frames. They look like ladders. Polyether, they have oxygens, these O's all over them, both backbones. Short backbone, long backbone. And they all look kind of like this. This is the general motif. Different derivatives of each one in this little part of the molecule. Each of these toxins is different. Each of them causes a different effect. Each of them has a different potency. When one measures the size of these molecules as they get airborne and blown ashore, the particles, we get a very nice distribution of aerodynamic diameter. And if you break out upper respiratory, middle respiratory, and then pulmonary function, how far these particles get down into your lungs, you'll see that in this area where most of the toxin particles are in this area of the upper respiratory where cough, irritation, and runny nose occurs. Perfectly correlates with human potency. Lower respiratory, as concentrations get higher and this peak gets taller, there's a little bit that spills over onto both sides. You start to develop wheezing and shortness of breath. Those are the kinds of times when people get scared because you can't catch your breath. Very serious is when pulmonary function becomes aberrant. Skipping inhalations, holding your breath, not able to exhale, diminished function for a variety of reasons. You don't actually absorb oxygen as well or release CO2. And then finally, pulmonary collapse, very unusual in Florida red tide. Very few healthy people die from Florida red tide. In fact, I don't think that there's even anything in the literature about deaths. Now it's going to get a little bit tougher here, and I want to. I'm going to step back so I can talk a little better. Can you hear me? Yeah. So what we're going to talk about, first of all, is looking at, this is a sheep inhalation protocol. These sheep are perfectly healthy. They don't hurt the sheep at all. They inhale toxins or therapeutics. They report to us on bronchoconstriction. They report to us on therapeutic intervention. These are used for asthma. They're used for cystic fibrosis. They're used for a variety of pulmonary diseases. And these are our babies. These groups of animals have been used for years and years and years. Okay, so we take very good care of them. We don't expose them to anything that's of real damage to them. We come up to where they start to have a problem and back away, just so that you're aware of that. And animal testing for all of that you don't want to do with it is important because the alternative is people getting hurt. And the last thing you want to do is get into a clinical phase 1 study and have people get hurt. Okay, so that's one of the reasons that we do this. Okay, so in the case of, so what we're looking at here is increases in bronchoconstriction. As you go up this way, it's increases in bronchoconstriction. As you go across this way, it's more cells. So this is Karenia brevis. This is the organism that causes Florida red tide. As you increase the number of cells, you can see that bronchoconstriction goes up. That's when these animals are inhaling particles of Karenia brevis. Okay? So that shows you that it is the organism that's causing the bronchoconstriction. If you extract the toxins from the organism and then correct the number of cells for the amount of toxin that's present in there and overlay them, you will see that in fact it is the toxins in the organism that are causing the bronchoconstriction. Are you comfortable with that? That it's the same lines. Okay. So that's bronchoconstriction, and that's by the Karenia species and then by the toxins that come out of the organism. So both of those are bronchoconstriction events. They also reduce tracheal mucus velocity. They slow down mucus. Well, they don't slow down production, but they slow down the expelling of the mucus, and then you swallow it generally to get rid of bacteria or other particles that are in your lungs. That's how you get rid of them. Or blow your nose, a variety of things. Okay. If one looks at 2 of the different toxins, one is called PBTX3, that's this one. You'll notice that with increasing time, the mucus velocity decreases and then slowly comes back. The most potent of these toxins here only decreases tracheal mucus velocity slightly and then plateaus off. And then vehicle alone doesn't really do anything. Vehicle is the stuff you dissolve it in. Okay. So if you have a high concentration of PBTX2, which is the most potent brevetoxin, along with a little bit of PBTX3, it gets into the lungs and it stays there because PBTX3 is inhibiting the tracheal mucus velocity for the bronchoconstriction. So it's a prolonged bronchoconstriction. Okay. So it's complicated. It's going to get more complicated because, in fact, if you look at all these toxins, they all have a different bronchoconstrictor effect. The PBTX2 we talked about in the last slide, 2.5 times more bronchoconstriction than at resting level, and all the other derivatives are less. So this is the really potent one. It's also the one that's present in the highest amounts in a Florida red tide. So that's why you have bronchoconstriction during a Florida red tide. So this is the first 5 years of the Florida Red Tide Program Project Grant. Okay, so after the first 5 years, we had studied and found all of the toxins that were present by the Florida red tide organism. Now you should understand that NIH grants are funded in 3 or 5 year increments. Ours was funded for 5 years. In 5 years, we had done all this work. And we kind of sat around and said, okay, what are we going to do for the next 5 years? Because in order to get more money, you have to write a competitive grant application to say, and here's where we're going next year. This is where the idea came up that anything that's in that organism, if the organism gets broken and up into the air, it's going to get blown ashore. So we said, okay, that's what we're going to do, and NIH bought it, and we moved forward. And the question then was, if there are other molecules that get airborne, what effects might they have on pulmonary health? Here's the predominant one that we found. This is Brevenal. It is a molecule that is about half the size of a brevetoxin, and it is a red tide toxin antagonist. If you have bronchoconstriction in an animal and you apply this material, the bronchoconstriction is resolved. It's therapeutic. This particular molecule, like the toxins, occurs in all Karenia isolates. It's everywhere. The ratio varies with culture conditions. So sometimes there's a lot of toxin, not much Brevenal. Sometimes there's a lot of Brevenal, not much toxin. If there's a lot of toxin, what do you have? Follow me. What do we have? Bronchoconstriction. If there's a lot of Brevenal, do you have bronchoconstriction? No. Okay. In the lab and in the field, Brevenal counters the toxic effects of the toxin. All the time, every time, no matter who does it. We had colleagues in other universities who were presenting material on non-toxic clones of the organism. And the first sentence would be, we have a non-toxic clone of Karenia, and it doesn't produce toxin. When you look at that clone and actually do the analytical measurements, it has a lot of Brevenal, and it doesn't have very much brevetoxin. In a similar fashion, super toxic, we have a really toxic clone by animal studies. You look at it analytically and say, well, it just doesn't have any Brevenal. And so really the Brevenal is modulating the toxicity. So the composite toxicity is always the result of toxins, metabolites, and Brevenal, the ratios of those. We have very high concentrations of cells in the water sometimes, no bronchoconstriction, sometimes no, very low concentrations, and everybody's coughing and sneezing. This helps explain why not all red tides have strong respiratory effects. That's a big one for NIH. Similarly, when you take Brevenal after you've induced a bronchoconstriction event and you add Brevenal, you can see that you can restore the normal pulmonary function to near baseline levels. You can do this either with a molecule that we create synthetically in the lab or actually a natural Brevenal molecule. So, what do these materials do? They reduce toxin-induced bronchoconstriction. We know that. They restore tracheal mucus velocity to pre-toxin levels. And they restore whole lung clearance to pre-toxin levels. And if you co-administer the Brevenal and the brevetoxin together, mix them together, and then give them to the animal, they reduce the effects. The bronchoconstriction is less. Now, this last experiment shown here in red is that you can prevent the toxin effects if you pre-treat with the antagonists. So if you pre-treat with Brevenal and then give toxin, there's no bronchoconstriction. Okay, it's a protection. Serendipity 2. The first one was having a toxin and an antitoxin in the same organism. It's actually a paradigm shift for the whole oceanographic field. This one actually gave Bill Abraham the opportunity to actually see what the Brevenal does in the absence of toxin. And he did this by using this particular molecule, this Brevenal molecule with the aldehyde on the end. It has a 4% bronchoconstriction effect itself. It is an antidote for the toxin constriction. The other one that you'll see on some of these slides is this beta-naphthalene brevetoxin that was produced by Kelly Ryan from FIU when she was a postdoc in the lab, analyzed by Sherry Parker and assisted by Lynn Fieber with Sherry's postdoc to develop another antagonist, but we won't talk very much about that tonight. Now this is a fun slide, and I'll talk you through this one. This is a really interesting one because what we're talking about is basically musical chairs. Okay? So think of this as a musical chairs event. How many of you played musical chairs when you were a kid? How many of you won? Exactly. Okay, so in this case, however, we are not going to be taking chairs away. Instead, what we're going to do is we're going to continue to add children to the mix. Okay? And we're going to give one child a red shirt. And everybody else is going to have a different colored shirt. This is that red child right here. A red child sits in that chair, the brevetoxin receptor, and these other children are going to attempt to displace that child from her seat. And those children, you can see that they look a little different than this child. They actually have a leg, so that when they sit in a chair, they go like this. So they may sit in this next chair, but they have their leg over another chair. What does that mean? The kid's not going to be able to sit down, right? So the kid loses the game. All right. When you do this experiment, this is the traditional receptor binding experiment, you will see that with increasing concentrations of children, it's more and more likely that the red-shirted child is not going to get a seat. That's the basis for drug therapies. You've just learned the basis for drug therapies. We could do all the kinetics, but really the third graders are better at doing that than adults are, so we'll let them do that every day. If you then change the study, instead of having red toxin, you have red antitoxin, red Brevenal. And you do the same study, you find that kids that are in the same family as the red-shirted child can effectively displace that child from the seat, but the other kids can't. And what this really tells you, these two experiments together, is that the Brevenal molecule and the brevetoxin molecule interact with different molecular targets. And the Brevenal target, the target that's the therapeutic, actually affects the ability of the other one to bind to its site. So the sites disappear. And that means those targets are gone, the toxin can't bind. That's the basis for this. Okay. In summary, this molecule physiologically, Brevenal, reduces the hyperresponsiveness of lung. It reduces bronchoconstriction, makes the lung totally compliant. It promotes mucociliary clearance. It promotes whole lung clearance. And Bill Abraham found as well that it prevents inflammation. Now, I don't know if you know that many cystic fibrosis patients that have the pulmonary aspects don't really die from the thick mucus, they die from the bacterial infections that occur as a result, or the scarring that occurs as a result of the persistent infections. This molecule counteracts all of that. So what's happened? This molecule is a new receptor, and that new receptor acts at lower concentrations than any other molecule that's been described to date. It promotes tracheal mucus velocity increases. It basically thins mucus by a mechanism we'll talk about. Promotes whole lung clearance, and by that, Bill Abraham called and said, you know, when I did the pre-treatment experiments, I think you have a cystic fibrosis treatment. Just that easy. And then he filled all the all the little boxes in. So it's a fractal bringing together of seemingly disparate discoveries to create this helpful therapeutic from a fluoride type, which is traditionally a pulmonary toxin. So how can Brevenal help in cystic fibrosis pathophysiology? First of all, you should understand that cystic fibrosis is the result of a specific ion channel, a channel through a membrane that promotes the transport of chloride ions. And chloride ions are negative. In a similar fashion, there are, and this is the outside of the cell, the inside of the cell. In a similar fashion, there are sodium ion channels that transport sodium from the outside to the inside. And the ability of these two types of channels, along with several other types, to open and close and maintain ion balances directly results in the ability of water to maintain homeostasis across membranes. And so in a case where a cystic fibrosis transmembrane regulator does not function properly, water cannot get out of the cell and is stuck inside. When water can't get outside to hydrate the mucus becomes drier, thicker, and ropier. And so many of the current traditional, or the current therapeutics that are being proposed or being developed are to attempt to change the functioning of this aberrant functioning CFTR. So the question is, are there other ways to circumvent the effects of the cystic fibrosis mutants without having to touch the CFTR? And that would be to modulate or correct all of the reagents or all of the membrane agents around and create other ways of getting water back and forth or chloride ions back and forth. And so in a normal channel or a normal CFTR situation, there's normal chloride transport, matches sodium, and everything works right. In aberrant chloride transport, chloride can't get out, sodium can continue to come in, that holds the water in, the water can't get out, mucus gets thick and rough. It's not just one kind of a mutant. CFTR has at least 2,000 different known mutations. Currently, the progress on therapeutics to correct CFTR is dependent on specific mutants. So if you think there are 2,000 known mutants, the worst case you have to have 2,000 different drugs to treat 2,000 different types of cystic fibrosis. Probably not, there are classes. There are generally 5 different classes. Either the DNA is messed up and it doesn't appropriately transcribe to get that channel into the membrane, or it doesn't make the protein correctly, or it has a mistaken amino acid which causes it to fold incorrectly, or it doesn't get traffic to the membrane correctly, or once in the membrane it gets broken down too quickly. All of these are classified as CFTR mutants. There are at least 5 different classes. You notice that the slides change from my black and white slides to color slides for Serendipity Pharmaceuticals. In 2013, Serendipity Pharmaceuticals licensed all of the polyketide and ladder frame polyether intellectual property, the patents and all of the use patents from UNC Wilmington, and are now developing them in collaboration with industry and with the Cystic Fibrosis Foundation. This summary slide is where the corporation is presently. They add to what I've already told you, that exacerbations of asthma, COPD, and CF are associated with increased levels of free neutrophil elastase. It's an enzyme in the airways. Inhaled human neutrophil elastase is a putative mediator of asthma, COPD, and cystic fibrosis, and it slows tracheal mucus velocity. HNE itself stimulates protease-activated receptor 2 release. So you have now a cascade from HNE to PAR2. If we take our Brevenal and try it in between the HNE and after the PAR2, we can actually show a resolution of the effects of each of these over the baseline here. This is the HNE, PAR2. This is the effect together with Brevenal. Together with Brevenal, this is a post-treatment. Create the effect and then relieve it. And then PAR2 is the second part of this cascade, and you can see that both amiloride and Brevenal are drug alleviate the effects of PAR2 on airway responsiveness. So within the cystic fibrosis therapeutic pipeline, I just pulled this down off of the web yesterday. These are the areas that current drugs are working. They're working in areas of CFTR modulation. They're interested in airway liquids. They're interested in anti-inflammatories and a variety of others you can see. Our drug Brevenal, we're in our first exploratory study of Brevenal in pursuit of the CF Foundation funding, where we expect to have a drug that will affect ion transport, restore mucus regulation, and reduce inflammation. This is the pipeline, how you get to the drug. We won't talk about it in general. Here's where we are right now. These are months at the bottom. We're in 12 months. This is 108 months to final approval. This is all the way through clinical phase 1s, clinical phase 2s, clinical phase 3s, pre-NDA meeting with the FDA, submit the NDA to get a new drug out on the market, and that's it. We started in 2007. We'll start the clinical trials probably in 2017, first quarter. Okay. So the translational phase of development of this drug has now begun. It is now out of university hands. Our university laboratories are preparing the material, providing it to the corporation, who will then develop the clinical studies. You can see these are two molecules. This is the beta-naphthalene we produced in the lab that I didn't talk about tonight. But the Brevenal is the important one. It's active at very, very low concentrations. Our renewable source is the cultures to produce the material now, but we also have natural product through a synthetic method, a zipperase activity from a group at MIT, and genomic activity from a group in the Craig Venter Institute in California. The efficacy that we demonstrate occurs at a new receptor site. Previously uncharacterized, it is a new locus for drug development. That's an important piece all by itself. The therapeutic index is very high, it's 20,000. That means you can go 20,000-fold above the therapeutic concentration and not see ill effects. Very good. Has potential therapeutics, as I mentioned already, for cystic fibrosis, COPD, and also for, believe it or not, neurotoxic shellfish poisoning from Florida red tide, or ciguatera fish poisoning due to tropical fish that people consume that have ciguatoxin in them. During this time period, UNCW has been granted 4 patents. We have a 5th pending, and the synthetic one in the laboratory, we have an additional 2 patents that are granted. All of these are licensed to Silurian at this point. So this is a success story. This is a translational science program that is complete. It's complete. Now the corporation takes over, and we look for a drug to be out there in the not too distant future. We're back to these again. I just want to talk a little bit about the Escortins, because I think they have really pretty pictures, and I think you'll enjoy the pictures. In this particular case, we were trying to develop assays, test kits, for neurotoxic shellfish poisoning. And using our little laboratory assays with just cell membranes, we got these wonderful curves that look a lot like these are the same, the same curves that I showed you in the past. However, we decided we could get a better idea of what was really going on if we used whole cells. And when we used whole cells, the faculty member came to me and said, uh-oh, we got problems. The color, the fluorescent color on the cells, or the fluorescent color associated with the toxin, goes into the cells. Uh-oh, serendipity strikes again. Hadn't expected that at all. Well, so, gee, this was a small company working with the university. I said, well, it'd be interesting. Why don't you take our different ladder frame materials, the 2 different brevitoxin backbones, and Brevenal, and couple them to that same green thing and tell me what you get. Do they all do that? Do they all carry this green color across? And you know, sure enough, Bo Dippy is this green element. Kind of looks like, what is it, like Halloween 3 or something? Bo Dippy, brevetoxin, plus a lysosome tracker. And if you track with different things, you can actually see where the yellow color shows up, where this molecule actually delivers inside the cell, and it looks like it all is in endoplasmic reticulum for this particular one. Okay, so that's one, that's Bo Dippy. Well, Bo Dippy is good, but how about another one? Does it work in different cell types? Take 9 different tissue culture cells and tell me if it goes into all of them. Answer is, yes, it does. Give me a different color dye that you attach to the brevetoxin. Do those go in? Yeah. Fluorescent tag doesn't make a difference. Yeah, they all go in. So then the big experiment. Take these, all of these different dyes that are all available. They all have different colors. Link them to toxin, link them to Brevenal, put them in cells. Let's see some pictures. And just for kicks, we threw cancer chemotherapeutic agent daunorubicin in one. Daunorubicin is fluorescent and should give us a really nice picture. So here are the pictures. Any cell that is colored outside of the blue nuclear stain has toxin or Brevenal linked to a fluorescent linker. The fluorescent linkers are plus minus charged. They can be negatively charged, positively charged, neutral, hydrophobic, fat soluble, water soluble, round, flat, free to move around, fixed in position. Everything that we link brevetoxin molecules to, or Brevenal molecules to, the CF drug, go across membranes. So there's another mechanism. The mechanism of transport of these materials has never been described before, and it's an active transport, so they can take up these materials against a concentration gradient, ideal for drug development. The problem is that both brevetoxin and Brevenal have pharmacologic effects. You don't want to be attaching drugs to things that already have drug-like activities, because you can produce all sorts of untoward side effects. So what I say here is what we needed was a non-toxic ladder frame polyether. And this is it. This molecule is called brevicin. We've never been able to find an activity for brevicin. We actually published the structure of it because it was so unusual, but didn't do anything. Big mistake on our part, but the uses are still okay. If you attach, either give Bo Dippy, the green one, alone, or attach Bo Dippy to brevicin, over a period of 60 minutes is the time lapse. Bo Dippy alone, green. Do you see any green color inside? Just little bits, huh? Link Bo Dippy to brevicin and watch the material go in. The same thing with daunorubicin. Daunorubicin is the cancer chemotherapeutic agent linked to brevicin. All by itself, without any brevicin, no uptake. Linked to brevicin, look at the uptake. Now you can even mix these two together. As long as they're not linked together, there is no uptake of the dye. So it is the polyether, the brevicin, that's carrying this material across the membrane. This is my most complex slide. How does this do this? I don't know. That's an artist's rendition. But these escortins bind to specific endogenous, meaning always there, transport systems, active transport systems, of both the clathrin and the caveolin-coated pit type. Basically what happens is the fluorescent material is engulfed by part of the membrane invaginates. As it invaginates, it carries the particles into the cell. The outer cell membrane is broken apart and the materials are released into the cell. The interesting thing is that although these are very similar to caveolin and clathrin-mediated uptake, and they respond to most of the inhibitors of those two types of uptake, there are characteristics that indicate that it is a different type of transport system that probably hasn't been described yet. Again, the molecules give us a new mechanism for uptake of materials into cells. So what are we doing with this one right now? Well, this moved a lot faster than Brevenal. It seems like the first time you do this, it takes forever, and then people know who you are. And so as soon as we started demonstrating this, we were approached by the cosmeceutical industry for the delivery of vitamin C in a brevicin coated material. That particular thing is in preclinical studies. They're going through all of their toxicology now. Even more important is the approach of using vitamin C linked to one of these escortins in wound healing. And this has the attention of both DARPA and the Department of Defense for battlefield, much in a similar way as superglue did in the Vietnam War. Gave you things that you didn't have before that can actually help you in a battlefield. And what this basically does is allow vitamin C to stay in touch with the collagen synthesis mechanisms that create the scars, the scarring to create and close the wounds. And then a third one is, it's been licensed for diabetes type 2. I have no idea what they're going to do for that. I don't think that that will work, but you know, a lot of people are interested. So in total, what I want to leave you with is that it takes big groups of people to do this. I don't care what anybody says, serendipity plays a big role in all research, particularly in the subjects that I've shown you tonight. And I guess what I'd say is if you feel a little bump in the road, oh, there's serendipity again, okay? It's 3 different groups that work together. The first group is my Mar Bionic group at UNCW, my research faculty members I work with, all of our technical staff, undergraduates who turn over about every year or so. They all get great training in the laboratory. And then unusual, I have a retired AstraZeneca executive who works with us, very useful. And I was fortunate to hire an intellectual property attorney, a patent lawyer, very early on in our work, so that we could come and talk to him and say, Steve, should we be protecting this? And he would evaluate it and say yes or no, and then we'd move forward. Very quickly, 24-hour turnarounds. And then my program project team. That team is the team that helped develop all of these paradigm shifts, all of these disruptive technologies that are becoming so useful. I mentioned already Tom Mende, who started me out with, I think we can make a dent here. The program project team, these are the leaders. There were actually about 28 people, that worked in this particular project. These are the leaders of the various cores. Richard Pierce from Moat, who did all of the airborne analysis. Barb Kirkpatrick, who worked with Laura Fleming on all the epidemiology. Laura is a physician. They were out swabbing people's throats and nostrils and doing pulmonary function testing. Young Sung Cheng, who's a very, very accomplished particle physicist, can helped us with the sizes of the particles. Janet Benson, who did all of our mouse work and the preclinical toxicology. Lori Baker from the CDC. We just couldn't have done it without the CDC's input. And then outside of Andrea Bordelais, who discovered the original molecules, Bill Abraham is irreplaceable from Mount Sinai Medical Center. When I first met Bill when the program project started, Laura said, you need to get Bill involved. I've never met anybody as smart as he is, and I like the way he thinks. She was right. And then finally, I'll just acknowledge that Silurian Pharmaceuticals is taking this into clinical trials. They are interacting with the FDA, and I would say look for some really good things from them in the future. The program was funded by the National Institute of Environmental Health Sciences. It is the only institute of NIH located outside of Bethesda. It is in Research Triangle Park, North Carolina, 2.5 hours from us. That NIEHS funding was supported by the Florida Department of Health as well as the Centers for Disease Control throughout the entire process. That is another type of partnership that is just immeasurably important to what we do. And I'll finish with telling you that we just suffered through Tropical Storm Anna that formed, I don't know, somewhere off of Palm Beach or something. Anyways, you didn't have to worry about it, right? Moved up toward us, dumped rain on us for 4 days, got really cold, then got really warm, and this is the result. All of my roses in 1 day are all in full bloom. I have neighbors from all over coming and picking the roses. Probably when I'm gone, they're taking them all. But anyway, first roses of the season, and with that, I will thank you for your attention.